Hello everybody and welcome to the endocrine system. This first video is going to be about hormones, the different types of hormones and some example pathways, as well as the different ways that we can secrete hormones. So we'll start with what is the fundamental question of the video. What are hormones? The hormones are your body's chemical messaging system. If this video were to be about the nervous system instead of the endocrine system, then what we would be discussing in that case are voltage-based signals. But here in the endocrine system, we're going to be counting on molecules binding to appropriate receptors and eventually causing some physiological changes. And as we'll come to see, there are many ways in which we can achieve this goal, as some hormones seek to take a more indirect approach to passing along a signal, and some seek to do it themselves with a more direct approach. Now with that said, there are definitely a few different ways that we can go ahead and secrete hormones, and we'll go ahead and discuss some of these now paired with visual examples. The first being endocrine hormones, which are released directly into the bloodstream. And we can see that in our visual example over here. We have our cell that is generating hormones, passing them along to the bloodstream. The hormones will make their way out of the bloodstream, eventually landing at receptors at the target cell. Next would be paracrine hormones, which are hormones that are secreted by one cell, and the target are neighboring cells. And we can see that in the middle example. We have the originator cell here generating some hormones. And here the geographic location is a little bit more prominent because this cell is a direct neighbor to the originator cell. These hormones will come on down and directly interact with the receptors of the target cell. Now, last but not least, we have what is maybe the most interesting mode of hormone secretion, and that is known as autocrine hormone secretion. Let's take it over to the visual. When we're talking about autocrine hormone secretion, we're talking about one cell that is generating hormones that will then come back and affect the very same cell, binding to receptors on the cell. And this makes the cells that generate these autocrine hormones very cool, because they're first of all capable of synthesizing these amazing chemical messengers, but they also house the corresponding receptors, meaning that they can directly benefit from the release of their own hormones. There are also a few main types of hormones that we're going to go ahead and dive into, and don't worry about jotting this down just yet. We're going to dive into each of these in a little bit more depth as we move forward. I just want to set a baseline here to start. The first one is known as peptide hormones. We also have steroid hormones and amino acid derived hormones. Now, when you think of peptide hormones, think proteins. P for peptide, P for proteins. These are protein-based hormones, meaning that they are made of polypeptides. Now, appropriately enough, the synthesis of these protein-based hormones occurs in the rough endoplasmic reticulum, which we can note right here. It sits just outside of the nucleus of a cell. Now, why is the rough ER the appropriate place to be generating these hormones? Well, if you don't recall, the rough ER is riddled with ribosomes, small structures responsible for aiding protein synthesis, and the rough ER as a whole is responsible for aiding protein synthesis. So we now know what peptide hormones are, but how do they do what they do? How do they go about passing along these signals? Well, remember, peptide hormones are proteins, meaning that they're generally going to be large and pretty polar. What this also means is that because of their polarity, they're going to be water soluble, which means they can freely flow through the blood. But because of that same polarity, they are going to be lipophobic. They are not lipid soluble, which means that these hormones cannot directly pass through a cellular membrane. So instead, they're going to have to take a different course of action. They're going to bind receptors on the cell surface so they don't ever have to cross over into the intracellular domain on their own. And so we have a great visual example over here on the right. Here's our mock peptide hormone in orange. It's in the extracellular environment, and instead of crossing directly through the cell membrane, which it's not capable of due to its polarity, it is instead going to associate and bind to a receptor on the extracellular domain. This is a process known as indirect stimulation, where a signal is still being propagated in the intracellular environment, but it's not the original hormone that's going to be doing it throughout the whole process. Instead, that peptide hormone is going to interact with a receptor on the cell's surface and kick off on the inside of the cell something known as a signal transduction pathway. And in just a moment, we are actually going to be able to see a couple of examples of different modes for how this indirect stimulation takes place. But before we go ahead and do that, I want to set a little bit of a baseline by nailing down some key terms that we should know, as well as giving a very generalized overview of the modes that we'll see in more depth later on. The first thing that we need a rock-solid understanding of is the ligand-gated receptor. These are those big receptors that we were talking about a little bit before that are responsible for grabbing onto that peptide hormone on the outside of the cell and making some signal transduction magic happen on the inside of the cell. Now, once the beginning of this process occurs, we've taken that peptide hormone and we're causing some kind of effect on the inside of the cell. Well, what happens next? We need more messengers if we can't rely on the hormone in the intracellular environment. And that is where the secondary messengers come into play. These secondary messengers are signaling molecules that are available in the inside of the cell, but they are not the original hormone. And so now we're going to go ahead and cover some very important examples that are going to come up in later slides when we start to dig into those different modes of ligand-gated reception and signal transduction pathways. We'll go ahead and start with CAMP, cyclic AMP. We also have IP3, which is short for inositol triphosphate, DAG, which is short for diacyl glycerol, and Ca2+, which is very simply calcium 2+ ions. And as promised, here are those two types of ligand-gated receptors that we're going to cover in a little more depth as we move on. The first is known as a GPCR, which stands for G-coupled protein receptor. The other form of ligand-gated receptor that we're going to cover is known as a receptor tyrosine kinase, or RTK. Now, while we're here, I also want to give a quick shout out to ligand-gated ion channels. Ligand-gated ion channels are liable to appear in ligand-gated receptor scenarios. What happens is we have this interaction with the ligand-gated receptor and our hormone on the outside of the cell. We have all of this secondary messaging happening inside of the cell, and sometimes those secondary messengers will act as ligands themselves. Recall, ligand-gated ion channels are protein channels that exist in a membrane which allow for the passage of ions that are too polar to pass through membranes themselves. Ligand-gated ion channels in particular respond to a physical, mechanical alteration when they bind a ligand. And so we are more than capable of achieving this result with a secondary messenger. Now here we have our first example of a peptide hormone and a signal transduction pathway. And before you start worrying and rushing and trying to get everything on this page down all at once, don't worry about it. We're going to go through each step a couple at a time, and they're going to be paired with some visual examples so we can really get into this and break it down. Feel free to come back to this slide at a later point in the video or in your own independent study time for review. Alright, so let's go ahead and kick off the IP3 DAG pathway discussion, and I'd like to preface by saying the reason it's called IP3 DAG pathway is that IP3 and DAG are two secondary messengers that are eventually going to be created in this pathway, and we'll get there. Don't worry about that just yet. And as always, I really like to keep the broader perspective in mind as we move through something as microscopic as this pathway. So as we're moving through each step, remember that this pathway at its core is about releasing calcium 2+ from the endoplasmic reticulum. So step 1, keeping in the spirit of peptide hormones, this whole thing is going to kick off when a ligand, in this case a peptide hormone, binds to our GPCR. And we can see that here. This is an expanded version of a visual that we've already seen. Here is our extracellular signal molecule, that peptide hormone, and it's going to associate with this G protein coupled receptor, remaining at all times in the extracellular environment. Remember, peptide hormones never cross directly through the cellular membrane into the intracellular environment. From there, the GPCR is going to go ahead and activate that G protein that is connected with in the intracellular environment. And in this case, this is going to occur by binding one molecule of GTP, a molecule that is very similar to the high energy molecule ATP. And so we can see that as well. We've had our signal molecule bind on the outside of the cell. On the inside, this translates to the activation of a G protein. So where do we go from here? As we said, G proteins will go on and do a whole bunch of important stuff for us inside the cell. In this case, for the IP3 DAG pathway, the G protein is going to go ahead and activate an enzyme for us, and that enzyme's name is phospholipase C, which can be found right in the center of our visual. Now, even just looking at the name of this enzyme, we can start to infer what its role might be, with the prefix being phospholip referring to phospholipids and the suffix ase referring to enzymes. In this case, phospholipase C is going to be something that cleaves lipids. We're going to go ahead and break down those ester bonds within lipids. Now, for the purposes of this pathway, phospholipase C is going to go ahead and target a specific lipid in the membrane known as PIP2, which we can go ahead and see right over here. And the cleavage of PIP2 is going to generate two separate secondary messengers, a couple that you've heard of already, IP3 and DAG. And here we can see our lovely DAG and IP3. Now here's where we kind of leave DAG to the side because it's going to go on to be very productive in other pathways. IP3 is going to carry on towards our calcium release goal. The secondary messenger IP3 is going to go over to the endoplasmic reticulum and directly bind a ligand-gated calcium 2+ channel on its surface. The ligand, of course, being in this case IP3. And so you can kind of imagine the creation of IP3 would be up here. It's going to go all the way down to the endoplasmic reticulum and bind to that channel. Once the ligand-gated channel is bound with IP3, it is going to open up and allow all of this calcium to flow out. And where is this calcium going to go? It's going to end up in the cytosol of the cell. Now what happens next is a perfect example of why signal transduction pathways are so efficient. Remember, we just started with one interaction between a peptide hormone on the outside of a cell and a receptor. Yet now somehow we have all of these free calcium ions that are going to go on to affect a bunch of different proteins that are going to go on to affect a bunch of different cellular responses. And we can really appreciate that when we take a look at the bigger picture. We're just starting with one little interaction over here, and our signal molecule doesn't even enter the cell, which is the most amazing part. With this pathway, we are able to turn one interaction on the outside of the cell into an activated protein inside, an activated enzyme, the creation of two secondary messengers, and the release of all of these ions that are going to go on to affect a whole bunch of different pathways. Alright, so now that we've really broken down the mechanism of a GPCR pathway, in particular the IP3-DAG DAG pathway, let's go ahead and talk about the other mode of signal transduction which is possible, the mode that uses receptor tyrosine kinases as their ligand-gated receptor. Receptor tyrosine kinases, or RTKs, are interesting because they're composed of two identical components that dimerize once they are bound by the appropriate ligand and activated, and you can kind of think of this very simply as two siblings holding hands, which is to say that the dimerization of the two components very simply means that they are going to come together and interact. Now when they do this, another thing that makes them very cool is they're going to cross-phosphorylate. Phosphorylation, of course, being the addition of a high-energy phosphate group to something, with cross-phosphorylation meaning that one component is going to phosphorylate the other, and vice versa. And so we can take a peek over at our visual example, and we're starting with inactivated RTKs, separated, not bound, and once we do bind our signaling molecule, they're going to dimerize and interact, and from there, they'll go ahead and cross-phosphorylate. These phosphate groups are then able to interact with other proteins, and this cross-phosphorylation gives rise to another secondary messenger response. We have reached the second type of hormone that we're going to discuss for this video, the steroid hormone. Steroid hormones are actually generated from modified cholesterol molecules. And if you look down to the visual, it's not hard to see the resemblance. So here is our initial cholesterol, and over here we have, for example, steroid hormones, testosterone, progesterone, estrogen, and aldosterone. The key identifying structure is the fused 4-ring structure. If we were to look over to cholesterol, we can see 1 ring, 2 rings, 3 rings, and 4 rings, with the 4th and final ring having a pentose shape rather than a hexagonal shape. And we can see that the same is true for our example steroid hormones. Let's look at testosterone. 1, 2, 3, 4 rings. Progesterone is the same, estrogen, and aldosterone. Now in terms of synthesis, steroid hormones are going to be generated in the smooth ER, the smooth endoplasmic reticulum, and steroid production in the smooth ER is just as appropriate as peptide hormone production in the rough ER, because the smooth ER is responsible for aiding lipid production, which is what we're working with steroids, because cholesterol, if you recall, is a lipid. And of course, we don't need to be working with the rough ER because we are not making steroids out of proteins. So who is making these steroid hormones? Who's going to be using their smooth endoplasmic reticulums to make these lipid-based hormones? Well, steroid hormones comprise all of the hormones that are released by the adrenal cortex and the gonads of humans. The gonads, of course, being the testes and the ovaries. For the adrenal cortex, the cortex being this outer portion of the adrenal gland, a gland that we're going to touch on in a later video, the important steroid hormones that it's going to go ahead and produce are glucocorticoids, mineralocorticoids, and androgenic steroids. For the reproductive organs, for the testes and ovaries, we're going to be producing progesterone, testosterone, and estrogen. Now it's over to the fun part. How do steroid hormones do what they do? Well, it's going to be a little bit different than peptide-based hormones. Because we're basing these hormones off of cholesterol, they are going to be lipophilic. not lipophobic, meaning that now with steroid hormones we have a means of directly crossing cellular membranes. So again, we can say that these are lipid-soluble hormones, but they are not water-soluble. So if we were going to transport steroids through the blood, for example, we would have to associate them with a water-soluble protein. What does it mean now that we can actually go ahead and cross the cellular membranes directly? It means that rather than taking an indirect approach where we require a signal transduction cascade, we can go ahead and jump right to direct stimulation, where our hormones are going to directly bind to receptors on the inside of the cell, and the hormone is no longer banished to an existence on the outside of the cell. And so once the steroids and the receptors come together, they will form something known as a steroid receptor complex, which will go on to directly affect DNA transcription, a process that is considered slow and gradual. So here we have another wonderful visual example that we can go ahead and work off of. We have our steroid hormones in the extracellular environment. Because they are lipophilic, not lipophobic, because we're using cholesterols to make these hormones, we can just go right on through the cell membrane. Once we are in the intracellular environment, our hormones are going to associate with a receptor molecule, creating this steroid receptor complex, and this complex will go ahead and bind to DNA and affect its transcription. And so here we have a mock mRNA that would result from the affected transcription of DNA based on the binding of the steroid receptor complex. And now once that mRNA is translated, we have a brand new protein that was the result of the signal passed down from the steroid hormone. I apologize if you're not typically one for real-world examples, but here's a quick one that might help us solidify our understanding of the comparison between steroid and peptide hormones. There's an old saying in soccer, let the ball do the work, which is to say that if you pass the ball down the field to your other teammates one at a time, rather than running it down the field yourself, you've let the ball do the work. You've avoided exhaustion, and you've avoided oftentimes physical encounters with defenders. But more importantly than anything, you've moved the ball down the field in the most efficient way possible. Peptide hormones go ahead and let the ball do the work. They will pass the signal along through different proteins, through different secondary messengers, and move the signal along in the most efficient means possible, maximizing the potential effects by using different components. In comparison, steroid hormones want to dribble the ball down the field. They prefer to be the quote-unquote flashy dribbler and do it all themselves. They want to go through the cellular membrane, into the nucleus, affect DNA itself. They want to do all the work, score the goal, and be the hero. The last type of hormone that we're going to discuss are amino acid-derived hormones, and they're actually pretty cool because they tend to have properties that are similar to both peptides and steroid hormones. Now, they're a little bit smaller than peptide hormones. Peptide hormones, remember, are made of proteins. Amino acids are the constituents of proteins. So we're not talking about full-blown polypeptides here. What we're talking about instead are hormones that are mostly derived from the amino acid tyrosine, which we can see right here. And so an example of a similar property that an amino acid-derived hormone might have to, say, a peptide hormone is the polarity. We're not going to be able to bring an amino acid-derived hormone directly through the cellular membrane because it expresses a similar polarity to that of a peptide hormone. And in terms of synthesis, we'll be producing these amino acid-derived hormones in the rough ER and in the cytosol of cells. And just like the way that the steroid has exclusive rights to the gonads and the adrenal cortex, all of the hormones that are made in the adrenal medulla, the other portion of the adrenal gland, are amino acid derived, as well as T3 and T4 hormones. And those hormones that are created in the adrenal medulla, something that we'll discuss in a little bit more depth as we move on in this series, are epinephrine and norepinephrine, your fight or flight hormones. All right, so at this point, let's go ahead and move on to a mini quiz to see what you've learned. Which type of hormone is secreted directly to neighboring cells? Go ahead and pause the video now, take a moment, and answer this question. All right, welcome back. The correct answer in this case is paracrine hormones. Remember, paracrine hormones are the ones that are going to be released, secreted by one cell, with the target being neighboring cells. Autocrine is incorrect. These are the hormones that are going to be secreted and then target the same cell that secreted them. Exocrine hormones are secreted by a mode that we didn't quite discuss in this video. They are secreted into glands for distribution, not related to what we're looking for in this question. And D, endocrine hormones are also not correct. These are ones that are secreted directly into the bloodstream. Okay, this has been Hormones, the first video on the endocrine system. I hope you enjoyed and learned something today. If you have any questions, feel free to reach out, but if not, happy studying. Hi everybody, and welcome to today's video on the hypothalamus and the pituitary gland. Today we're going to talk about both the hypothalamus and the pituitary, breaking the pituitary gland down into its two main constituent parts, and then we're also going to briefly touch on the pineal gland. So let's go ahead and begin by talking more generally about the key structures at play here, the hypothalamus and the pituitary gland. If you recall, the hypothalamus is part of the limbic system in the nervous system. It's a more centralized set of structures in the brain, with the hypothalamus existing right in here as we have zoomed in by our visual. And the hypothalamus has some very important roles, both related to the overall function of the nervous system as well as in relation to the endocrine system. So let's go ahead and start breaking down some of those. First of all, in terms of the endocrine system, the hypothalamus is going to be charged with the production and secretion of some very key hormones. And we'll see as we move along through this video how the hypothalamus and the pituitary gland coordinate activity to make this production and secretion as effective as possible. From there, the hypothalamus is going to be charged more broadly with the coordination of the body's internal environment and maintenance of homeostasis. Now, moving on to the pituitary gland, also referred to as the hypophysis, this is an extension off of the hypothalamus, and it actually can be broken down into two main parts, as we'll see, the anterior and posterior pituitary gland, each formed of different materials. One, as we'll come to see, is composed of neural tissue, and the other is composed of glandular tissue, with this differentiation and composition providing a wonderful degree of separation of responsibility. And we can see it in the visual, but we might as well go ahead and clarify that the pituitary gland, in terms of geographic location in the brain, rests just below the hypothalamus. So let's go ahead and begin by talking about the posterior pituitary, which is also referred to as the neurohypophysis. The reason it's known as the neurohypophysis is because the posterior pituitary gland is going to be the subset of the pituitary gland that is composed of neuronal tissue rather than glandular tissue. And so as a result, the posterior pituitary gland is a direct neural extension from the hypothalamus. And this structural relationship is actually very much tied to the function of the posterior pituitary gland. As we mentioned in the last slide, the hypothalamus is responsible for the release and production of certain hormones. When we're talking about the posterior pituitary gland, we're talking about a structure that's going to store some of these hormones that are produced by the hypothalamus, and then it will go on to be the structure that actually is responsible for releasing these hormones. So an important thing to remember then as we move along is that the posterior pituitary gland is not producing its own hormones. It's receiving hormones that are produced by the hypothalamus, and it is responsible for releasing those. And the two key hormones that we're going to talk about, made in the hypothalamus, released by the posterior pituitary, are antidiuretic hormone, otherwise known as ADH, and oxytocin. So first, we're going to go ahead and discuss antidiuretic hormone, or ADH, also known as vasopressin. Now, if you recall the chapter on the excretory system, the functional unit of the kidney is something known as a nephron. And it's this whole convoluted structure, as we can see in this visual to the right, with each different part of the structure having different functions in reabsorption and minerals and ions and water and all these different things. But the structure in particular that we're going to be focusing on for our purposes is the final one, the collecting duct. The collecting duct is involved in reabsorption of water via structures known as aquaporins, water channels. And what antidiuretic hormone is going to do for us is it's actually going to allow us to increase the amount of water reabsorption that the nephron is capable by increasing the overall number of these water channels, these aquaporins. And this is wonderful for us because we are now going to have a higher capacity for water retention and reabsorb more water back into the blood. And yes, of course, antidiuretic hormone is an antidiuretic. Now, why does this make sense? Not even speaking on the fact that antidiuretic is present in the name. Let's more so think about the function. The prefix anti is something that we're probably pretty familiar with, and in this context, we can think of it as referring to something preventative. And the suffix is probably more familiar than you might think. The suffix in this case is diuretic. It sounds like the word diarrhea, an affliction that, unfortunately, I'm sure many of us are very familiar with. So without getting too in-depth, let's start to think about how these two words, diuretic and diarrhea, might give us the ability to unpack what an antidiuretic hormone would do. The thing that's going to give us this answer and tie both together is the presence or absence of water. If an excretory deposit were to be made by a human that contains a large volume of water, this is something that we can immediately recognize as potentially synonymous with a diarrhea-like scenario. But let's dig a little bit deeper into that. What would it mean if the excretory product had a large volume of water? It means that we're not retaining water efficiently or at a high level. So then what do we have with antidiuretics? when antidiuretics are present, we should expect the opposite scenario to occur. We should expect an excretory product with a low water content. And we know that this is what antidiuretic hormone does because we already know that it increases the number of aquaporin channels and helps us reabsorb water in the collecting duct of the nephron of the kidney. Next, we have the hormone oxytocin. There are two main functions that we should talk about. First of all, we're going to target the uterus to stimulate uterine contractions during child labor. Next, oxytocin is also going to target the mammary glands separately from the uterus, stimulating the release of milk during breastfeeding. Now, what makes oxytocin such an efficient means of achieving both of these is that the way it operates in both cases is via a positive feedback loop, and we have actually an example on the bottom right of your screen. So before we dive into it, let's just imagine that over here we have the hypothalamus. It's producing oxytocin. It gives it to the posterior pituitary gland, and now we're actually releasing oxytocin into the environment. In this case, we're talking about stimulating contractions during childbirth. So here is our contraction indicator. And the way that this is going to work is that the contractions actually serve as the positive indicator for the production of more oxytocin. So more oxytocin equals more contractions equals more oxytocin, which is clearly a very beneficial setup when you're trying to deliver a live child. All right, so that was the posterior pituitary gland. It's time to go ahead and move on to the anterior pituitary gland, also known as the adenohypophysis. Adenohypophysis, of course, being because the prefix here, adeno, is in reference to glands, and the anterior pituitary gland is, of course, constructed of glandular tissue this time rather than neural tissue. And the most important thing about the anterior pituitary gland, the thing that distinctly separates it from the posterior, is that it's going to go ahead and produce its own hormones, a function that is made possible by the glandular tissue composition. But saying this does not mean that it's completely distinct from the hypothalamus in terms of function. It still relies on the hypothalamus to be stimulated and release the hormones that it produces itself. And so we actually have two separate categories of hormones that the hypothalamus is capable of releasing in order to stimulate the anterior pituitary gland one way or the other. The first is known as hypothalamic releasing hormones. If one of these hormones is to reach the anterior pituitary gland and stimulate it, we're going to release the hormones that the anterior pituitary gland generated. Hypothalamic inhibiting hormones, on the other hand, are the exact opposite. If one of these hormones were to reach the anterior pituitary gland from the hypothalamus, the result would be the inhibition of release of the hormones produced by the anterior pituitary gland. There is, however, one more key detail that we need to discuss. In the case of the posterior pituitary gland, the mode of transfer of hormones from the hypothalamus to the posterior pituitary was relatively straightforward. Both were composed of neural tissue, with the posterior pituitary gland serving as the direct extension of the hypothalamus. The anterior pituitary gland does not have that luxury, so instead we're going to count on something known as the hypophyseal portal system. Because if we can't rely on a direct connection between these two structures, what we're going to need to do is pass the stimulating hormones from the hypothalamus into the bloodstream and send those down to the anterior pituitary. Using our regular artery and vein system would see these hormones traveling throughout the body, passing through the heart and back up to the brain, just to reach the anterior pituitary gland, which geographically is located right next to the hypothalamus. Lucky for us, our body is more crafty than that, and we're going to utilize portal veins, which are very aptly named because they serve sort of as a portal from one part in the circulatory system to the other, bypassing the rest of the circulatory pathway. And so this is an extremely efficient means of connecting the anterior pituitary to the hypothalamus, despite the fact that one is not the direct neural extension of the other like we have in the case of the posterior pituitary. And so, without further ado, I present to you the stimulating hormones that are produced by the hypothalamus, pass through those portal veins, and target the anterior pituitary gland. The first goes by the shorthand GnRH. The long form is gonadotropin releasing hormone. And once it reaches the anterior pituitary and goes ahead and stimulates it, it will cause the release of two hormones, luteinizing hormone, LH, and follicle-stimulating hormone, known as FSH. The next is TRH, or thyrotropin-releasing hormone. When it interacts with the anterior pituitary, causes the release of something called thyroid-stimulating hormone, or TSH. We also have CRH, corticotropin-releasing hormone, and once this reaches the anterior pituitary gland, it's going to cause the release of adrenocorticotropic hormone, or ACTH. And last but not least, we have GRH, known as growth hormone releasing hormone, which appropriately enough, when it finally reaches the anterior pituitary gland, will cause the release of growth hormone, or GH. All right, so we've completed phase one when it comes to the anterior pituitary gland. We've released those hormones from the hypothalamus, and they finally reach the anterior pituitary, stimulating the release of its own hormones. There's actually two kinds of hormones, though, that are produced and released by the anterior pituitary gland. The first is known as tropic hormones, and these are going to seek to, quote-unquote, keep the ball rolling because tropic hormones target other endocrine glands with the goal of promoting further hormone release, with the goal of causing those endocrine glands to release their own hormones. Direct hormones, in comparison, take a little bit of a different approach. Rather than targeting endocrine glands and trying to stimulate the release of more hormones, they're just going to go right to the organs and try and cause some effects themselves. Now, at this point, you're probably wondering what's going on with this sad pig over here that seems to be crushed by all of his schoolwork. Well, actually, what's going on here is a very important mnemonic, flat pig. And I just want you to keep in mind flat pig as we move along. It'll make a lot of sense later. Let's kick things off with a couple of tropic hormones. The first that we're going to discuss is FSH, or follicle stimulating hormone, which actually has two separate functions, one in females and one in males. In females, it's going to stimulate the growth of the follicle, and in males, it's going to go ahead and mature sperm. Another important anterior pituitary gland tropic hormone is LH, or luteinizing hormone, which stimulates many different things. It's going to stimulate ovulation, the formation of the corpus luteum in females, and the production of testosterone in male gonads. Now, there are actually a couple of kind of silly ways that you can remember both of these separate from the flat pig example that we were talking about before. FSH kind of looks like the word fish, if you were to swap out the letter I in fish for just nothing. And fish, as we all know, swim. So does sperm. So if you're looking for a little trick to remember what FSH does, you can remember that FSH sounds like fish. Fish swim, it matures sperm. Sperm swim as well. There's also one for LH, luteinizing hormone. If you want, you can use LH as an acronym for large and hairy. Two adjectives that would become more pronounced with more release of testosterone. And in the visual example down below, you can see how these tropic hormones are really pushing for the release of other hormones rather than going ahead and causing these effects themselves. Here in the middle, we have the anterior pituitary releasing luteinizing hormone and follicle stimulating hormone. On the left, we have a very simplified pathway for males where we're targeting the testes and the release of a different hormone, testosterone. And on the right, we have an equally simplified pathway for females where we're targeting the ovaries and the release of estrogen and progesterone. Another tropic hormone released by the anterior pituitary gland is ACTH, or adrenocorticotropic hormone. ACTH, once released, will go ahead and affect the adrenal gland, and it will cause the adrenal gland to release glucocorticoids, which are very important because they allow us to do a little stress combating. They are also going to go ahead and increase the glucose levels in the body. And the final tropic hormone that we're going to go ahead and discuss is TSH, or thyroid stimulating hormone. And appropriately enough, TSH is going to go ahead and stimulate the thyroid gland to release some hormones, and the names of those hormones are T3 and T4, with the eventual goal with T3 and T4 of increasing metabolism. And so in our example to the right, we can see just how this cascade takes place. We begin in the hypothalamus with TRH, or thyrotropin releasing hormone, which will stimulate the anterior pituitary gland to release TSH in this example, or thyroid stimulating hormone. And instead of causing an effect on its own, it's going to stimulate another endocrine gland, which we'll actually discuss in a later video, the thyroid gland, to release its own hormones known as T3 and T4, and eventually we'll start to work towards that goal of increasing metabolism. Now, how about the direct hormones from the anterior pituitary gland, the ones that are actually going to go right to the organs and try and cause some changes themselves? The first is known as prolactin. Prolactin is in fact going to go ahead and stimulate the mammary glands and increase the production of milk. Now, this is going to sound familiar. It's going to sound very similar to oxytocin, but there's some very finite differences between the two. Prolactin, first of all, increases milk production. Oxytocin stimulates the release of milk. Second of all, prolactin shows up following childbirth and does not occur in a positive feedback loop scenario. Oxytocin shows up during breastfeeding when release of milk is necessary and does operate in a positive feedback loop. The other direct hormone released by the anterior pituitary is known as growth hormone, otherwise known as somatotropin. Now you can probably guess the function of growth hormone. We're going to be targeting body cells and looking to grow and divide those body cells. And it's finally time to return to our good friend the flat pig and get an explanation on why it's so important. Now that we've seen all of the tropic and direct hormones, it should start to make a little bit more sense. Flat is a mnemonic for all of the tropic hormones, and pig is a mnemonic for all of the direct hormones. And looking throughout the list, we can confirm that, yes, of course, this is the case with FSH, LH, ACTH, and TH all being tropic hormones of the anterior pituitary, and prolactin and growth hormone or somatotropin being direct hormones. And then I is just kind of thrown in there as a place filler to make the mnemonic sound a little bit better, so don't worry too much about I. And so as always, it's very good to keep the broader perspective in mind as you're working through some of these more narrow pathways. And so here's a visual that ties together all of the hormones released by both the anterior and posterior pituitary and relating it to the original release of hormones by the hypothalamus that kicks off everything. This way you can really appreciate the different pathways that are possible. We can go the route of the anterior pituitary and stimulate it to release its own hormones, which we've talked about. We have over here the tropic hormones, and on the right here the direct hormones, which go right on to affect organs, while the tropic hormones seek to affect other endocrine glands. We can also go the direct neural connection route, focusing on the posterior pituitary, which will house hormones produced by the hypothalamus, which we can see here that only have direct functions. And one more quick note that we do have for you today is about the pineal gland. And the pineal gland is very important because it produces something that might sound very familiar. It produces melatonin, with melatonin, of course, being an incredibly important hormone for regulating your circadian rhythm in sleep patterns. All right, let's go ahead and do a mini quiz and see what you've learned. Which of the following hormones causes the release of milk during breastfeeding once released? Read this question carefully. Go ahead and pause the video now. Take a minute and answer this question. The correct answer is B, for oxytocin. Remember, oxytocin is one of those hormones released by the posterior pituitary and acts in a positive feedback loop. More breastfeeding equals more oxytocin. More oxytocin means more release of milk, which encourages more breastfeeding. More breastfeeding encourages more release of oxytocin, and so on. A, prolactin is incorrect because this is a hormone released by the anterior pituitary that stimulates the production of milk, not the release. And it also occurs following childbirth and does not act in a positive feedback loop. It's not going to be C, follicle stimulating hormone. Remember, FSH, follicle stimulating hormone, is more so associated with the development of the follicle in females and sperm maturation in males. And luteinizing hormone is also incorrect. Remember, this is a tropic hormone, which down the line is going to seek to cause things like ovulation and the release of testosterone. All right, this has been the hypothalamus and the pituitary gland. I hope you took something away from this video. If you have any questions, please feel free to reach out. But if not, happy studying. Hello everybody, and welcome to today's video on the thyroid and parathyroid. We're going to be touching on both glands as well as the hormones that each secretes, and we're also going to be touching on some relevant disorders. Now, the thyroid and parathyroid glands by definition are endocrine glands, which means we're going to be looking to secrete some hormones. And we'll go ahead and begin with the thyroid gland, which is actually the largest of all the endocrine organs. And as we can see in our visual to the right, it sits just in front of the trachea and is represented by this larger, vaguely H-shaped structure. Now, in association with the thyroid gland are the parathyroid glands, these little structures here that we can highlight on our lower visual, and you can actually see them in this visual as well. Notice as the model rotates, the posterior side of the thyroid gland becomes visible, revealing the location of the parathyroid glands. But the true functions of each will be revealed as we dig a little bit deeper into the released hormones. So let's go ahead and talk about the thyroid gland first. The three important hormones released are known as T3, T4, and calcitonin. And we're going to go ahead and begin by talking about T3 and T4 first, better known as triiodothyronine and thyroxine. And immediately what you might recognize is the visual on the bottom right, because this was present in a prior video when we were talking about the hypothalamus and the anterior pituitary gland. And if you recall, the basic mechanism for release of T3 and T4 is the release of thyrotropin-releasing hormone from the hypothalamus, which will stimulate the release of thyroid-stimulating hormone, very appropriate, from the anterior pituitary, which will go on to stimulate the thyroid gland to release T3 and T4. And what's very crucial when discussing T3 and T4 is distinguishing between the things that make them similar and very different. When it comes to things that make them similar, let's start with their overall role. Both of them are responsible for increasing metabolism. We're going to look to increase basal metabolic rates in the body. And we can actually reference our visual example one more time to get a bit of a better idea of how this might occur. They're going to achieve this by influencing gene expression throughout the body. As we mentioned, both T3 and T4 are going to be released from the thyroid gland in response to the release of TSH from the anterior pituitary gland. They are both in fact derived from the amino acid tyrosine, which you can remember as the 3 T's, and both of them have very important negative feedback functions for TSH, thyroid stimulating hormone, as well as TRH, thyrotropin releasing hormone, which goes on to stimulate the release of TSH. Now it's time to break into the things that make them different. We'll start with T3. T3 is actually considered the activated form between T3 and T4. Structurally, this is because it has one less iodine atom, but the result of this small fundamental change actually makes T3 4 times more potent than its counterpart T4. Now looking over to T4, in comparison, T4 is considered the more stable form, and therefore if you were to just take a random sample in the blood, what you would see is that T4 is the more abundant form because it is more stable. With more stable meaning in this case that it has an additional iodine atom. Well, how about that third hormone that the thyroid gland releases, calcitonin? Let's go ahead and talk about that now. Calcitonin will actually go on to decrease blood calcium. And a decent way of remembering calcitonin's function is that calcitonin sort of sounds like calcium toning, which sort of sounds like toning down the calcium. And I know there was a lot of steps there, but I promise if you follow it all the way through, you'll never forget what calcitonin does. But what does the decrease of blood calcium levels in the blood actually look like? It looks like the stimulating of osteoblasts and the inhibiting of osteoclasts. The stimulating of osteoblasts being so crucial because those osteoblasts, the cells that are going to look to build new bone, are going to take calcium out of circulation and use it to build that bone. Whereas osteoclasts would be looking to break bone down, which in a scenario where they are active would free up some calcium, but since we are inhibiting those osteoclasts, the calcium is going to stay bound in the bone. Separate from targeting the bone, calcitonin will actually also go out to the kidneys and the intestines to decrease calcium reabsorption. Unfortunately, there are some disorders that can come along with the thyroid if things don't go as planned. The first that we're going to go ahead and talk about is hypothyroidism, with hypothyroidism being the undersecretion of both T3 and T4 from the thyroid. And since we know at this point that T3 and T4 are going to look to increase metabolic rates, the undersecretion of both will reduce our metabolic rates. The counterpart to hypothyroidism is hyperthyroidism, or the oversecretion of T3 and T4. In this case, we're going to be increasing basal metabolic rates, but we're going to be having a hyperactive or overactive thyroid gland to achieve this. And unfortunately, we can actually go even deeper than that because both hyperthyroidism and hypothyroidism are possible precursors to something known as a goiter, with a goiter being an irregular enlargement of the thyroid gland, visible in our visual example down below, with both normal and enlarged thyroid glands depicted. But we know that hypothyroidism and hyperthyroidism are kind of the antithesis of each other. So how do they both achieve the same outcome, the production of goiter? Well, it actually occurs by two separate modes. When we're talking about hypothyroidism, the underproduction of T3 and T4, we will in fact end up with low T3 and T4. But recall that the beginning of the process to secrete T3 and T4 is the release of TRH from the hypothalamus. And so our brain is going to try and compensate, but we're going to end up overcompensating and oversecrete TRH. And this is going to lead to an enlarged thyroid gland from hypothyroidism. Now, in the case of hyperthyroidism, remember we have an oversecretion of T3 and T4 giving us high levels of T3 and T4. These, as we know, are released by the thyroid gland. An overproduction of T3 and T4 is going to give us a hyperactive thyroid gland, which we discussed in a previous slide, which will give us the same enlarged thyroid gland. Now, what's interesting to note is that iodine deficiency. Recall that iodine atoms are one of the main constituents of T3 and T4, is the leading cause of goiter formation. And last but not least, let's take a look at the important hormone that is secreted by the parathyroid glands, which is known as parathyroid hormone. And parathyroid hormone is actually the antithesis of calcitonin. It's going to look to increase calcium levels in the blood. And we'll run through the function list. It's going to look very similar because it is actually the exact opposite of what calcitonin does. We're going to stimulate osteoclasts instead of osteoblasts, breaking down bone and releasing free calcium. We're going to inhibit the osteoblasts, so we're not taking any of that free calcium and putting it into new bone. And we're going to increase calcium reabsorption in both the kidneys and the intestines. And so here we have a visual example where we can see both pathways, and you can really start to appreciate how both parathyroid hormone and calcitonin do the exact opposite things. All right, so at this point we can go ahead and move on to a mini quiz to see what you've learned. Which of the following is an active hormone that is released by the thyroid gland? Go ahead and pause the video now, take a moment, and answer this question. And the correct answer in this case is B, T3. Remember, T3 is the activated form between T3 and T4 with one less iodine and being 4 times more potent than T4. We can then go ahead and cross off CT4, which is known as the stable or more abundant form. We can cross off oxytocin because this is not something produced by either the thyroid or the parathyroid gland. It's actually produced by the hypothalamus and released by the posterior pituitary gland. And parathyroid hormone is released by the parathyroid gland, not the thyroid gland. All right, this has been the video on the thyroid and parathyroid glands. I hope you enjoyed and took something away from this video. If you have any questions, please feel free to reach out, but if not, happy studying. Hi everybody, and welcome to today's video on the pancreas. In this video, we're going to distinguish the different parts of the pancreas by their tissue types, and we're also going to talk about the different products that each tissue type produces. The first question then is, what is the pancreas? Since it shows up in the endocrine system chapter, you can probably guess that it's an endocrine gland. But what distinguishes the pancreas from all the rest is the fact that it is composed of both endocrine and exocrine tissues. If you recall from a prior video, we broke down these first 3 listed modes of hormone secretion, with endocrine being the mode where hormones were released from one cell directly into the bloodstream and released later on in the circulatory system at a target cell. There was one that we've been saving until just now, and that is exocrine tissues, which we can see right here. So where endocrine cells would release hormones directly into the bloodstream, exocrine cells by comparison release their hormones into ducts. And this is demonstrated for us in the visual down below. We have this mock lining of cells in this U shape here, and they're all producing hormones, shipping them into this tube-like vessel known as a duct. And as we'll see, this gives the pancreas a wonderful array of capabilities since it is able to utilize both of these modes. So now that we understand the difference between endocrine and exocrine, let's break down the specific endocrine and exocrine tissues of the pancreas. For the endocrine tissues, otherwise known as the islets of Langerhans, we're going to be focused on secreting peptide hormones, in particular, a few that you may recognize: glucagon, insulin, and somatostatin. And as we move on, we'll break each down by their role, but I want to begin by letting you know where each comes from specifically. For another great thing about the pancreas is that its endocrine tissues are separated into different types of cells, which we can see in both visuals to the right, granting these endocrine tissues a great degree of separation of responsibility. When we're talking about glucagon, these are hormones that are going to be produced by alpha cells, which we can note right here. In comparison, insulin is going to be released by beta cells instead. And the final peptide hormone released by the islets of Langerhans is somatostatin, which is released by delta cells, which we can highlight right here. Now over to the exocrine tissues. Now remember, with the exocrine mode, we're going to be utilizing ducts. And if you recall from a different chapter, the chapter on the digestive system, we actually touched on the pancreas for reasons outside of hormone release, because what the pancreas is going to use this exocrine mode of release and these ductal systems for is the release of digestive enzymes into the small intestine. And if you have already watched that chapter on the digestive system, then the following should seem very familiar. We have the protease zymogens, trypsinogen and chymotrypsinogen. We have an enzyme to break down carbohydrates, pancreatic amylase, and we have bicarbonate ion, which is very basic to balance out the highly acidic gastric juices. These are all released from the pancreas through their pancreatic ductal system, inundating the pancreas in the above system. Once these enzymes leave the pancreas, they'll actually go into a duct that is common between the pancreas and the gallbladder and funnel down into the small intestine. All right, so let's go ahead and discuss a pair of those peptide hormones released by the endocrine tissues, glucagon and insulin, and they sort of have dueling purposes as they're both trying to solve glucose-related issues, but they're both solving opposite issues with opposite effects. So we'll go ahead and start with glucagon, which we know at this point is released by the alpha cells. Glucagon is going to show up when we have low blood glucose. And so what should we do in times of low blood glucose? Well, there's plenty of options to go ahead and tackle this issue, but the pancreas's favorite is asking other tissues to break down their glucose stores, specifically the liver and adipose or fat tissue. So glucagon from the pancreas will stimulate the liver and fat tissues to break down their glucose stores, freeing up that glucose and effectively increasing the blood glucose. On the flip side, we have insulin, which we now know is released by beta cells. And herein lies the opposite nature of insulin and glucagon, because insulin will be released in times of high blood glucose. And so now we're trying to solve a different problem where we have excess glucose in the blood. The solution for the pancreas then is to stimulate different tissues around the body that store this glucose to go ahead and take some out of the blood. In particular, the liver and adipose tissue, like we discussed with glucagon, but also muscular tissue is going to be tasked with storing glucose when it is stimulated by insulin. So how does this transfer of glucose actually take place? We know at this point that insulin is a peptide hormone, incapable of crossing cell membranes themselves, which means that it's going to have to interact with a ligand-gated receptor. In insulin's case, it's going to opt for a receptor tyrosine kinase. So insulin will bind the RTK in the extracellular environment, causing it to dimerize and become activated on the intracellular environment. Intracellular environment of the RTK, as we know, will result in the stimulation of secondary messenger molecules, and these specific secondary messenger molecules are going to stimulate glucose transporters. In particular, we're going to be increasing the number of glucose transporters on the cellular membrane. And we can see that effect in our visual. We have the increasing of these transporters, which can then take glucose from the extracellular environment, in this case the blood, and bring them into specific cell types that we're targeting, in this case liver, fat, and muscle cells, so that they may be stored in these cells. So what about somatostatin? Why didn't we include it in the other slides about insulin and glucagon, even though we clearly had it grouped in a prior slide and know that it's produced by the same endocrine tissues? Well, the reason is the functionality. Somatostatin has a bit of a different function from insulin and glucagon, and it is an inhibitory hormone. And so where insulin and glucagon kind of have this opposite relationship, trying to solve a similar but still opposite problem, somatostatin is actually going to inhibit the release of both insulin and glucagon. So if somatostatin is actually released by the pancreas, more specifically the delta cells, as we see down here, then what we will have is the inhibition of the release of insulin from beta cells, effectively halting the increase of glucose stores in liver, fat, and muscle tissues, the inhibition of glucagon release from alpha cells, which will inhibit the breakdown of these same stores and the release of free glucose into the blood, and it will also inhibit the release of a hormone that we saw from the anterior pituitary gland, growth hormone. And so here's one more slide. I don't want to spend too much time here. You can kind of pause it at your own pace or come back to it at a later time for studying purposes, but I do want to take a moment to appreciate that we're adding some more hormones into the big picture here. We've already talked about our hypothalamus and our anterior and posterior pituitary glands, and we've also covered the parathyroid and thyroid glands. But now we're starting to introduce some hormones responsible for the regulation of blood glucose levels from the pancreas. All right, so let's go ahead and do a mini quiz and see what you've learned. If blood glucose levels are determined to be sufficiently low by your body, which of the following is a potential response? Go ahead and pause the video now, take a moment, and answer this question. All right, welcome back. The correct answer in this case is D, the release of glucagon from pancreatic alpha cells. Recall that glucagon will target fat and liver cells and ask them to release their glucose stores in times of low blood glucose so that we may increase that blood glucose level. Answer choice B, the release of insulin from pancreatic beta cells, is the opposite. When we have too much glucose in the blood, we're going to ask the muscle cells, liver cells, and fat cells to take some of that out of the blood and store it. Calcitonin from the thyroid gland is incorrect because it has nothing to do with regulating glucose and instead shares a relationship with osteoblasts and osteoclasts to regulate calcium levels. And C, somatostatin from the delta cells, is also incorrect because this is an inhibitory hormone that actually regulates the release of both insulin and glucose. Okay, this has been the video on the pancreas. I hope you learned something today, and I hope you enjoyed it as well. If you have any questions, please feel free to reach out, but if not, happy studying. Hi everybody, and welcome to today's video on the adrenal gland. Today we're going to break down the adrenal gland into its different parts, as well as discuss the different hormones released by each part. And so before we get too deep into things, I want to talk about some more general anatomy, including where could we find the adrenal gland in the body. The adrenal glands are a little bit small, so we've highlighted them in the visual to the right, and we actually have two separate adrenal glands, both of which can be found on the superior surfaces of the kidneys which they associate with. What superior is in reference to is being closer towards the top of the body or the head. And so what you can see in this small portion of the visual, as well as the bigger visual to the right, let's highlight the adrenal gland associated with the kidney over here, is that the adrenal gland is associated with the quote-unquote top of the kidney. Now within the adrenal gland, we actually have two distinct structural categories that we need to differentiate now because they're responsible for producing different hormones. The first that we'll discuss is known as the adrenal cortex, which can be seen right down here. This is indicative of the outer region of the adrenal gland. And so if we have highlighted the outer region, you could probably guess that the adrenal medulla is to be indicative of the inner region, and we can highlight that here and confirm that that is true. Now, overall, both the adrenal cortex and the adrenal medulla and the adrenal gland as a whole is responsible for combating stress. Now, how exactly both the cortex and the medulla go about this is something we're going to dig deeper into as we move along in the video, but I want you to keep this general idea in the back of your mind as we move forward. All right, so let's go ahead and start differentiating between the cortex and the medulla in a little bit more depth. We're going to go ahead and begin this conversation by talking about the adrenal cortex. And right away, we're going to go ahead and tap into that theme of stress combating. The adrenal cortex in particular is responsible for combating longer-term stress. So how does the adrenal cortex actually go ahead and do this? Well, if you remember back to a previous video, we discussed the adrenocorticotropic hormone, or ACTH, and its release from the anterior pituitary gland. In our discussion of ACTH, we actually talked about how the main purpose of ACTH coming from the anterior pituitary is to stimulate the adrenal gland. And once we actually see this stimulation occur, the adrenal cortex will go ahead and release steroid hormones in particular, which will make a little bit more sense as we move on. Now with this, there are actually two main steroid hormone types that the adrenal cortex is going to release that we should differentiate and discuss now. The first are known as glucocorticoids, or colloquially referred to as cortisol. And glucocorticoids, or cortisol, is going to do a couple of important things for us. First of all, it's going to start to break down some glucose storages, freeing up glucose and raising blood glucose levels, giving us long-term access to glucose, which can be broken down as a fuel source. And in particular, we're going to be targeting liver, muscle, and adipose, or fat cells, and asking them to break apart these glucose stores. The other important thing that glucocorticoids are going to do for us is lower our immune response. Now, immediately upon hearing this, it may sound a bit scary, but really what we're going to be able to utilize this for is reducing long periods of inflammation. And this function is the reason why you may have actually heard of the word cortisol. And that is because this is a steroid that is used in medicine to combat inflammatory diseases. Moving on now from cortisol, we also have mineralocorticoids, otherwise known as aldosterone. Now, the main function of aldosterone is to go ahead and increase our blood volume, but it goes about it in a very interesting way. We're not just going to start pumping water into the blood, and actually, the functionality of aldosterone doesn't even directly target water. Instead, we're going to be targeting sodium ions, Na+. By targeting Na+ and increasing its reabsorption, we will cause a scenario where the solute ratios in the blood are beginning to change with an increase of sodium ions. And what happens if the concentration of solutes or ions increases on one side of the membrane? The answer, of course, is the establishment of a gradient. And so the water is going to follow these sodium ions back across the membrane into the blood via osmosis, evening out the gradient and increasing our blood volume. The adrenal cortex is also capable of producing small amounts of androgens, male sex hormones that we're going to discuss in more detail in the next video. And now we can go ahead and move on to our discussion on the adrenal medulla. Where the adrenal cortex is responsible for combating long-term stresses, the adrenal medulla is responsible for dealing with short-term stress. And the way that the adrenal medulla is going to go about doing this is a little bit different than the way that the adrenal cortex functions. First of all, we're going to be relying on input from the sympathetic nervous system. And if you've already gone ahead and watched the section on the nervous system, then the sympathetic nervous system may sound familiar. The sympathetic nervous system, for review, is part of the autonomic or automatic function nervous system that contributes to our fight or flight response, providing effects like the vasodilation of blood vessels in the skeletal muscles and the increase of heart rate. So before we even move on, do we see a link here? We should. We're linking your fight or flight response, which has to do with responding to immediate stressful environments, and a part of an endocrine gland in your body that is responsible for dealing with short-term stress. And before we move on to talking about the specific hormones produced, I want to mention that these are amino acid-derived hormones. But as we start to get into these hormones, it should be no surprise leading off that catecholamines, otherwise known as epinephrine and norepinephrine, otherwise known as adrenaline, have to do with your fight or flight response. Catecholamines can be thought of as your fight or flight hormones. And as we mentioned, the effects of these catecholamines are going to be things associated with aiding this fight or flight response. Things like increasing your heart rate, getting more oxygen up to your brain and to your muscles. Another example would include the breakdown of glycogen, freeing up a bunch of glucose and giving your body more resources to break down for energy in this quick, instant, stressful scenario. Now, it's worth noting here that epinephrine or adrenaline acts more throughout the body, and noradrenaline or norepinephrine acts more as a neurotransmitter. And to make some of these magical effects happen to help us with our fight or flight response, epinephrine is actually going to bind to some special receptors, known as either alpha or beta adrenergic receptors. And in particular, the association of epinephrine with these receptors can either cause vasoconstriction or vasodilation. Recall here that vasoconstriction would be indicative of a shrinking in diameter of a blood vessel, allowing less blood to flow through at a given time, whereas vasodilation in comparison would be indicative of the expansion of diameter to allow more blood to flow through at a given time. Now, the vasoconstriction would occur if epinephrine were to bind to the alpha receptors, and vasodilation would occur then if the epinephrine were to interact with the beta receptors. Now, I have a way for you to memorize this. It's kind of funky, but bear with me. If you find yourself in this immediate stressful scenario that triggers your fight or flight response, your alpha or first response may be to freeze up, which we can think of as constriction. However, as time goes on and you start to analyze the scenario, your beta or secondary response may be to open up and tackle the situation, otherwise known as dilation, sort of an expansion of self, as dilation is an expansion of diameter. So now it's time to talk a little bit more about the differences between long-term and short-term stressors, and I really want to get into why steroid hormones make sense as the response to one, and amino acid-derived hormones make sense as the response to the other. So let's go ahead and start with long-term stresses, which at this point we know are combated by the adrenal cortex. Examples of a long-term stress would include prolonged and insufficient access to food and water. Now, short-term stresses are more immediate, and we know at this point they are combated by the adrenal medulla. And examples could include anything that fits into the sudden frightening event category, something that's going to make you feel like you are in immediate danger. So now I ask the question, why? Why are steroid hormones released by the adrenal cortex to handle long-term stresses? And why are amino acid-derived hormones released by the adrenal medulla to handle short-term immediate stresses? It has to do with the hormone solubility. Steroid hormones, as we know from a prior video, are lipid-soluble but not water-soluble, which means that they're capable of crossing membranes themselves and directly affecting DNA transcription. On the other hand, amino acid-derived hormones share the polarity issue with peptide hormones. They are incapable of crossing cell membranes themselves and must interact with a receptor on the extracellular environment in order to kick off a signal transduction pathway on the inside of the cell. And this makes amino acid-derived hormones lipophilic and not lipid-soluble. And it is this discrepancy in solubility that causes one set of hormones to be selected for one role and the other to be selected for a different role. Signal transduction pathways can get more done quicker because on the inside of the cell, you are producing multiple secondary messenger molecules that can go on to affect multiple different pathways all at the same time, triggering a domino effect in each pathway that can generate more secondary molecules that can go on to stimulate more effects and more pathways. Steroid hormones are a little bit less efficient because the effects are one-to-one. The steroid comes through, binds the receptor, makes that steroid receptor complex, goes ahead and binds the DNA itself, and affects transcription. But because the signal transduction pathway can get more done faster, it is the choice to handle short-term stresses, which is why we have catecholamines as our fight or flight hormones. All right, so at this point, let's go ahead and move on to a mini quiz to see what you've learned. What is one function of cortisol? Go ahead and pause the video now, take a moment, and answer this question. Okay, welcome back. The correct answer in this case is answer choice C, lowering the immune response. Recall that cortisol can be used to fight inflammatory diseases because one of the functions of cortisol is lowering long-term inflammation. Answer choice A, increasing blood volume, is incorrect. This would be a function more indicative of aldosterone or the mineralocorticoids. Cortisol is never going to act as a male sex hormone. Those are known as the androgens. And the binding of alpha receptors and the causing of vasoconstriction is something that we would more think of as a role for epinephrine, not cortisol. Okay, this has been the video on the adrenal gland. I hope you enjoyed and took something away from this video. If you have any questions for us, please feel free to reach out, but if not, happy studying. Hi everybody, and welcome to the final video in the endocrine system series. Today we're going to be talking about the testes and the ovaries, as well as some hormones that are common to both, and how these common hormones have differing effects based on whether we're talking about the testes or the ovaries. We're also going to discuss feedback loops, including positive and negative. So first, let's do a bit of recall. How are we getting hormones to the testes and the ovaries in the first place? If you remember from an earlier video, the endocrine gland that's going to go ahead and stimulate both of these is the anterior pituitary gland. If you recall even further, the hypothalamus is going to release a hormone known as gonadotropin releasing hormone, or GnRH, that's going to stimulate the anterior pituitary gland to release some hormones that will go on to stimulate both the testes and the ovaries. And that is because the hormones in question are tropic and will seek out other endocrine glands. Now, the hormones in question for this video are going to be luteinizing hormone, or LH, and follicle stimulating hormone, or FSH. Both LH and FSH are going to make their way to both the ovaries and the testes, but as we'll see, they have differing effects. For example, in the ovaries, we can generally say that we are going to see the production of estrogen and progesterone, while the result in the testes will be the production of testosterone. Alright, so let's go ahead and discuss the ovaries first. We'll start the conversation with luteinizing hormone. At a certain point in the menstrual cycle, what we will see is a surge of luteinizing hormone, which will kick off ovulation, the releasing of the egg from a structure in the ovary known as the follicle. What we'll see as a result is once the follicle releases the egg, it will go on to form a different structure known as the corpus luteum. The difference, of course, between the follicle and the corpus luteum being that the follicle is the fluid-filled sac that houses the egg, and the corpus luteum is the structure that's left behind once the follicle deposits the egg in ovulation. But another reason why the corpus luteum is very important is because it's also capable of producing the hormones estrogen and progesterone. So now we can go ahead and move on to follicle stimulating hormone, or FSH. We're actually going to rewind the clock a little bit in the menstrual cycle for FSH, because once it shows up, we're going to see the growth of the follicle, which we know at this point is the structure that comes before the corpus luteum that actually houses the egg. And as the follicle matures, it will also go on to produce estrogen and progesterone. And so what's really nice about LH and FSH in the ovaries is if you're trying to think about how to remember what each does, all you have to do is look right to the names. Let's start with luteinizing hormone. Let's circle the first word, luteinizing. Luteinizing hormone is responsible for the corpus luteum. Sounds very similar to luteinizing. Follicle stimulating hormone is equally appropriate because follicle stimulating, let's highlight follicle, is responsible for the growth of the follicle. Now in the testes, let's go ahead and begin with luteinizing hormone. When we see stimulation of the testes by luteinizing hormone, the main thing that we can think of is the production of testosterone. And this is a process that occurs in a specific type of cell in the testes known as the Leydig cell. So, are there any quick and easy ways to remember that luteinizing hormone causes the production of testosterone? Of course. Remember that luteinizing hormone can be shortened to LH, and testosterone is capable of making someone large and hairy, which also can be shortened to LH. So luteinizing hormone, LH, can make someone large and hairy. Think testosterone. Follicle stimulating hormone, or FSH, has a different role and actually targets a different cell. With the follicle stimulating hormone, we're going to be targeting the maturation of sperm cells, and the cell of the testis in question for the maturation of sperm is the Sertoli cell. And if you remember, there is a little memorization tool for follicle stimulating hormone as well. FSH looks like fish if you were to take out the I, and fish swim, so do sperm. So when you're trying to think about what follicle stimulating hormone does in the testes, think about FSH, fish swim, and so do sperm. It is time to talk feedback loops. We're going to begin with positive feedback loops. When you enter a positive feedback loop, the resulting product will actually go on to stimulate the production of more of the stimulus. That's why we're going to go ahead and highlight positive feedback loop green for go ahead, make more. And you can kind of think of a positive feedback loop as a hamster on the wheel. The more the hamster runs, the more the wheel turns. The more the wheel turns, the more the hamster has to run to keep up with the wheel. And again, the more the hamster runs, the more the wheel turns, and so on and so on and so on. Both the hamster running and the wheel turning increase the presence of each other. Negative feedback loops are more common in nature, and this is quite the opposite of a positive feedback loop. In a negative feedback loop, the product is actually going to inhibit the production of the stimulus. A good example would be hunger. Your body tells you you're hungry, so you make and eat food, but the eating of that food doesn't make you more hungry generally. It usually tells your body, okay, enough is enough, that instinct is shut down, and you're good. But let's look at a couple of more applied examples when we're talking about hormones. So, for our positive feedback loop, we're going to use oxytocin from the hypothalamus and eventually the posterior pituitary gland and its relationship with contractions during childbirth. During childbirth, oxytocin will be produced, stimulating contractions. However, as these contractions continue, they will actually stimulate the body to make more oxytocin. The stimulation of more oxytocin will stimulate more contractions, and the contractions will stimulate more oxytocin. For our negative feedback loop example, we're going to talk about FSH and LH and their relationship with progesterone and estrogen in the ovaries. So as we know, an increase in follicle-stimulating hormone and luteinizing hormone is going to give us progesterone and estrogen by a couple of different means. But once we get to that luteal phase and the corpus luteum is formed once the egg is released from the follicle, both estrogen and progesterone are going to go on to inhibit gonadotropin-releasing hormone, the hormone released from the hypothalamus, and the one that stimulates the anterior pituitary gland to release follicle-stimulating hormone and luteinizing hormone in the first place. If we don't have any GnRH, then we can't have FSH or LH either. So in this case, we can clearly see how a product was formed, and then that exact product went on to inhibit the production of the stimulus. All right, so at this point, we can go ahead and move on to a mini quiz to see what you've learned. Which of the following is responsible for causing ovulation during the menstrual cycle? Go ahead and pause the video now, take a moment, and answer this question. Okay, welcome back. The correct answer in this case is A, a large surge of luteinizing hormone in the ovaries. Remember, we're going to count on estrogen from the ovarian follicle to enter a positive feedback loop with the production of luteinizing hormone, giving us a large surge of luteinizing hormone stimulating ovulation. B is incorrect. We're never really going to see a large surge of follicle-stimulating hormone the way we see it for luteinizing hormone. And even if we did, FSH is more concerned about the development of the ovarian follicle and not stimulating ovulation. C is incorrect. Even though the formation of the corpus luteum does have to do with luteinizing hormone, it comes after ovulation and is not the cause of it. And D, the release of an egg from the ovarian follicle, is incorrect because this is ovulation and it's not the cause of ovulation. Okay, this has been the testes, ovaries, and feedback loops. I hope you enjoyed and took something away from this video. Thank you for sticking around. This has been the Endocrine System. If you have any questions, please feel free to reach out, but if not, happy studying.